Nowadays, there are still many conditions for which there is no available cure, and patients must carry on with their lives with treatments that only mitigate some of their symptoms. IBDs or inflammatory bowel diseases are an example. These are long-term conditions characterized by the chronic inflammation of the digestive tract. Unfortunately, they are increasingly common, and the available treatments are not effective for many patients. Therefore, over the last years, more and more research has focused on improving the lives of these patients. The New Deal project is such an example. For 4 years, we have worked to develop a new technology with the potential to become a new treatment for IBDs and strived to bring it from the bench to the bedside. Our approach to reduce the inflammation consists of 2 elements: a molecule that has proven its efficacy in switching off the inflammation triggered by the gut cells, and, as this is a fragile molecule, a good partner for its delivery, a small particle that can transport it and protect it. These molecules are then combined in a final formulation that allows for their administration to patients with IBDs. For any therapeutic candidate to move from the laboratory to the clinic, many rounds of experiments, safety tests, and refining are required. We've proven the efficacy of our therapy in vitro and worked to ensure that it works with the added complexities of living organisms. For example, one of the things we must guarantee is that the active component of the drug arrives unaltered to the target cells through the digestive system. As the gut is a frontier between our body and the outside, it is prepared to hold back pathogens and other harmful substances. There are different barriers that serve as protection, but they also pose a challenge for the delivery of our drug. Fortunately, we count with different experiments to test if our therapy can overcome these obstacles. First, we can test the drug under variable conditions that reproduce the multiple harsh gut environments. Second, we use models that mimic the human digestive system and allow us to check whether our therapy successfully crosses the physical barriers along the way. The results of these experiments allow us to adjust the properties of our technology to achieve a successful delivery. Once our therapy has reached the area where our target cells are, we also must assure that it gets inside them to locally block the inflammatory response. To prove this, we use cultured cells and other models that better reproduce the physiological conditions and bring our tests a little closer to reality. We can analyze the penetrability of our therapy in clusters of human cells that resemble organs called organoids. We also count with animal models that are essential to ensure the safety of our therapy, as well as to test its efficacy in living organisms. With the samples that we recover from these experiments, we can examine what is happening at the cellular level. For this, we rely on a technology called flow cytometry that allows us to analyze the cells individually. To do so, first we add fluorescent markers to our samples that specifically bind to the 2 components of our drug. Then, we make the cells pass one by one through a laser and see whether the whole complex has entered the target cells or if any of the components has been lost along the way. As we can analyze cells from different organs, this technique also allows us to study the distribution of the drug throughout the organism and to rule out whether it is accumulating at undesired sites that could cause complications. All these experiments, together with the numerous toxicology and safety tests that we perform throughout the project, are essential steps that provide the required evidence about the potential and security of our therapy before moving into the clinic. In New Deal, we are fortunate to have practitioners and researchers on our team that allows us to be in contact with the clinic and answer to the real needs of IBD patients. The results obtained throughout our project aim at providing a solid proof of concept that facilitates the transition of the therapy to the clinical stage, setting the ground for future clinical trials. Clinical trials are composed of different phases ranging from tests in small groups of volunteers during the first phases, up to thousands of people in the final ones before its market authorization. These trials allow us to answer questions related to the potential side effects of a drug, its effectiveness to reduce or stop the disease, as well as its compatibility with available treatments. If the answers are clear, with a solid foundation, then the study is successful and the drug can be commercialized. Although the process that a drug follows until it reaches the market is long, all the steps are necessary to ensure that a therapy is safe and effective. Whether the results of the experiments are positive or negative, they always help us to know which path we should follow. Each experiment brings us a little closer to finally find an alternative therapy for IBDs, a solution that will improve thousands of patients' lives.